Hello, everyone. Welcome to the webinar, When Does Aging Begin? It's part one of a three-part series. The webinar is being recorded and will be posted on the GSA website. A notice to all attendees will be distributed once the recording is available. A question and answer session will immediately follow the live presentations. But throughout the program, we will be accepting questions through the questions feature accessible on the GoToWebinar panel. Without further delay, I'm going to hand the microphone to Dr. Ron Kohansky.
Thank you, Judy. As you can see, these are the three people involved in today's presentation on when does aging begin, which is a fundamental question whose answers, and there may be many answers, will help inform all aspects of research, clinical practice, and healthcare policy as these touch on aging populations. Of course, researchers will have a range of answers to this question, but even reaching the same answer, there might be different reasons for that conclusion. Today, we begin to explore this topic with presentations from two investigators whose work touches directly on this question, Dr. Daniel Belsky at Columbia University and Dr. Vadim Gladyshev at Harvard and Brigham and Women's Hospital. Next slide, please. For the discussion section, which will follow the two presentations of 20 minutes each, and then there will be 20 minutes for discussion, including some commentary back and forth between our speakers. But please add your questions to the Q&A box that's available, and we will read them as we have the opportunity. They'll follow the second presentation. Thank you very much. And now, Dr. Belsky.
Sorry, Ron, for the slight hiccup there. I think everyone should now be able to see my title slide. One quick note, I'm appointed in the Department of Epidemiology at Columbia in addition to the Aging Center. And a spoiler alert, the title I've given is the title of this session, When Does Aging Begin? But I'm not actually going to answer that question, although I think we'll be fortunate to hear something that comes closer to a legitimate answer from Vadim in the next talk. I'm instead going to provide a bit of an overview of how I think about this question from the perspective of epidemiology and the science working to link the biology of aging with public health. Okay, just a quick acknowledgement. Obviously, the National Institute on Aging, some other funders, my collaborators on this work, my mentors, Terry Moffat and Avshalom Caspi at Duke, Andrea Baccarelli at Columbia, Zhu Gao, now at Peking University, and Elliot Tucker-Drob and Paige Harden and their postdoc Laurel Raffington at the University of Texas at Austin. This probably goes without saying for most of you who are in attendance, but I want to orient us to what I'm going to talk about at least, which is the idea of aging as a biological process and specifically as the gradual and progressive decline in system integrity that occurs with advancing age. We understand this process now as a root cause of many different chronic diseases that afflict humans as they age, and a cause that arises from an accumulation of molecular changes at the level of our cells. And I've noted two prominent reviews on the bottom of this slide, and I'll leave it to the basic scientists who've developed these models to speak more about the specific causes of aging. But it's worth noting that these molecular changes are many, and they touch on both changes to the DNA within our cells, to the epigenetic marks surrounding those DNA, and to the products of our cells as well. And also that these many different hallmarks or pillars of aging are connected with one another, as illustrated in the pillars of aging figure on the right-hand side of the slide. And that encourages us to remember that changes occurring in one of these hallmarks or pillars are likely to manifest in other hallmarks or pillars as well. This is my cartoon model of the geroscience hypothesis, which motivates much of this work. The idea, again, is that these molecular changes that are accumulating at the cellular level feed forward to drive decline in the integrity of our multiple organ systems, which in turn give rise to a process of functional decline, disability, and ultimately death. And the idea, of course, of the geroscience hypothesis is that direct interventions on these molecular changes can forestall the decline in system integrity and delay or prevent functional decline, disability, and give us more healthy years of life. With that model in background, we can think about when it is that this aging process starts. And I like to orient folks when I talk about this to my thinking when I first encountered the field of aging, which was that aging was something that happened to people at the end of their lifespan, that we lived for a while in good health. There was this program process of development that fed forward into a healthy middle life period, and then we aged as characterized by the onset of various chronic diseases, disability, and ultimately mortality. And we've known for quite some time that who gets sick and when at the end of their life is importantly structured by experiences from the very beginning of the life course. So the work of David Barker and his colleagues on in utero determinants of later life health, now under the heading of the developmental origins of health and disease, have illuminated a number of pathways through which exposures in utero and in the first years of life influence our risk for disease, disability, and death at the other end of the lifespan. And what drew me into the field of aging was the idea that we could understand the process of biological aging as a critical mediator linking early life exposure history with disease, disability, and death, and ultimately generate new insights about pathways to prevention and intervention. That then led us to try and observe this process. As epidemiologists, it's awfully hard to study something by inference. The question was, is it possible to observe this process of aging in real time in humans, and in particular, in humans who are still young enough to be free of chronic disease? So moving this idea of aging back from the classical gerontology of people in their 7th, 8th, 9th decades of life into the middle period of the human life course. And this approach was guided by, again, this idea of aging as a gradual and progressive decline in system integrity. And therefore, the rate of aging, the rate of these biological changes accumulating at the level of our cells may be inferred from the rate of decline occurring across multiple different organ systems. Changes occurring in any particular organ system might reflect a specific disease process or some acute insult from the environment, but changes observed across multiple systems are more likely to reflect this common cause, which is this biological process of aging, or this set of biological processes of aging. And critically, if in fact these accumulation of cellular level changes are driving this decline in system integrity, we should be able to observe changes at the level of organ systems already in young, apparently healthy adults who are still free of chronic disease. And so our first study now 5 years ago assembled data from the Dunedin longitudinal study, a unique cohort that's followed about 1,000 individuals, all born between 1972 and 1973 in Dunedin, New Zealand, across the first 4 decades of their life at the time of this study. And from the time they were 26 years old, so everybody's born in the same year, they're all the same age at baseline. And again, when they were 32 and 38 years of age, 18 different measures of the integrity of various organ systems throughout their bodies were measured in parallel, and we were able to study how each of those indicators changed. We did observe evidence of a gradual and progressive decline in system integrity in this young healthy cohort. That's what you're seeing in the middle of the slide. The y-axis is scaled so that higher values correspond to values that indicate risk for disease or decline in organ system function. So you're seeing that on average in the cohort, system integrity is in fact declining across this period from the mid-20s to the late 30s. And then on the far right of the slide, you're seeing the measure we developed, which was based on individual differences in the rate of change in each of these 18 indicators. So for each of the 18 biomarkers, we modeled the rate of change and identified for each person their particular rate of change on each biomarker. Those were then composited to form this single measure, this pace of aging, which you see on the right-hand side of the slide, scaled in terms of years of physiological change experienced per 12-month calendar interval. And so most of the folks are aging at about 1 year at a time. Some are aging perhaps as much as twice as fast as that typical rate. Others appear to be aging not at all, meaning their physiologies when they were 38 years old look basically the same as they did 12 years earlier when they were 26. In this young healthy cohort, it wasn't possible to test whether this particular measurement forecast morbidity and mortality. These participants were still too young, but we were able to borrow measurements from the field of gerontology to investigate how faster change in physiology corresponded to declining function on the many capacities that we need to live healthy and fulfilling lives as we age. And so we observed evidence that people whose bodies were changing more rapidly showed poor physical function, poor cognitive function. They rated themselves as being in poorer health, and their faces looked older to undergraduates. So the question then becomes, how do we think about this measurement of aging in the middle of the life course or the end of the beginning of the life course, relating backward in time to exposures accumulating from the very earliest stages of life? And this is where my thinking was very strongly influenced by the Gavrilovs, from whom I learned a lot about the measurement of aging, in particular the idea that measurements of aging are quite different from measurements of age. And so that's one of the motivations for looking at rate of change in organ system integrity as our primary indicator of the aging process. But in this case, the Gavrilovs had introduced me to the idea that we could think about this gradual and progressive decline in system integrity as reflecting a progressive accumulation of random damage. And I'm sure we'll hear more about that in the next talk as well. But critically, early accumulations of damage structure the rate of future deterioration. And this is something that I think is known in the field of mathematics and engineering, and the Gavrilovs have adapted it to inform the understanding of biology of living things. And you can see in the figure they produced on the right-hand side of the graph, this idea that higher levels of damage produce aging rates. Here, it's the failure rate on a log scale, that are much more rapid. So if you early in life are accumulating more damage, you will exhibit a faster pace of decline as you approach young adulthood, midlife, and ultimately old age. And we put this hypothesis to a test in a paper published again a couple of years ago, where we assembled records from Dunedin study participants of their exposure to early life adversity. We know something about familial genetic load for longevity from the lifespans of their grandparents. We know about family socioeconomic circumstances in childhood, and we know from prospective records about their exposure to a range of adverse childhood experiences. And each of these are related to a person's future pace of aging. Those with longer lived grandparents tend to age more slowly. Those who grow up in poorer families or suffer more childhood adversity tend to age more rapidly. And on the bottom row, you see observations relating the integrity of the child in biological terms at baseline, their physical health, their brain integrity, their psycho-emotional integrity, measured here as an index of self-control, forecasting the rate of aging measured between 26 and 38 years. The next step with this pace of aging measure was to take it out of the Dunedin study so that it could be used to test relationships with late life morbidity and mortality, and also relevant to the topic of this session, to project it backward in life to investigate variation in the pace of aging in young people. And to do that, we turn to DNA methylation, which again, I imagine many of the attendees are familiar with the epigenetic clocks. It's been known for quite some time that DNA methylation marks, these are chemical tags on the DNA sequence that in classical genetics regulate gene expression. They change as we age. And in fact, it's possible to build remarkably accurate models of chronological age from these DNA methylation marks. And beginning with the work of the Eidecker lab and of course, Steve Horvath, there has been effort to develop indices of biological aging from DNA methylation. And so that's what we did in this study that came out earlier this year. We modeled that phenotype of longitudinal change in organ system integrity, pace of aging, from DNA methylation data to derive a novel index of the pace of biological aging, which we named, not very creatively, the pace of aging in methylation, and we appended to the front of that, so that's PoAm, the name of the study in which the index was derived, because it's quite possible that as we expand this approach into other cohorts, there may be variations that are specific to place or time. And so this is the Dunedin study-based pace of aging measured in methylation. And I'll walk you through a little bit of the validation data and then share with you some of the evidence that's been generated about how this index reflects on the early origins of aging. Okay, so just as a first look, we were interested in establishing that people who we measured to be aging faster, in fact, experienced a higher risk for mortality. This has often been the initial criterion validity check for measures of biological aging. And these are data from the normative aging study. We're here at the end of the lifespan. They have a mean age of 77 years. They've been followed up over nearly 2 decades with almost complete mortality in the cohort. And those who at baseline were measured to be aging faster according to this DNA methylation index of the pace of aging were in fact at increased risk for mortality. So you can see their survival plotted in red. This is the faster aging group in the cohort, slower aging group plotted in blue. We've recently done some additional work. Now we're going to step back in the lifespan just slightly by about a decade. These are data from the Framingham Heart Study offspring cohort, again showing that folks who are aging more slowly, this in this case indicated in the red, have substantially reduced risk for mortality as compared to those who are aging more rapidly, shown in the green here. And again, the pace of aging measure from DNA methylation is a continuous index, and here we've just created bins for the purposes of graphing survival curves. These are groups defined as slow agers, those with pace of aging and methylation, standard deviation below the mean, and the fast agers with pace of aging and methylation, standard deviation or more above the mean. And we see somewhat larger effect sizes here for what is about 10-year mortality in folks who are on average in their mid-60s at baseline. Taking a step further back in the life course, and again, here returning to the Dunedin study, we looked again at the same functional indices we'd studied in our original paper to ask if a methylation measurement of the pace of aging implemented when the study members were 38 years old could forecast their functional integrity 7 years later when they were 45 years old. And what you see in the slide is, in general, the answer is yes, although this particular methylation index was not associated with grip strength measured in these midlife adults. The other indices did show the expected correlations: faster aging, poorer physical, cognitive, and subjective functioning. And in fact, as the scatter plots are illustrating, evidence of functional decline from the age of 38 to 45 years in those measured to be aging more rapidly at age 38 from their DNA methylation. So this is supposed to be a talk about when does aging start. So I'm going to take you a bit farther back in the life course to have a look at some of those early life determinants that we know are related to risk of chronic disease and mortality later in life. And what we see in these data from the eRisk longitudinal study is that as of age 18 years, young people who grew up in lower socioeconomic status families and who experienced victimization as children show evidence of a faster pace of aging at the onset of young adulthood. And so this is sort of the point in the lifespan when I had initially been thinking about aging as beginning. We can think of a process of program development that is orchestrated by our genetic code that builds a reproductively mature human. And once that program has been completed, once we have reproductive viability, we can think of the process of aging as beginning in earnest as reproduction competes with somatic maintenance to maintain the organism versus projecting genes into the future, and things begin to fall apart. But in fact, as the Gavrilov's theoretical models suggest, damage accumulation begins far earlier in life than this. And may structure the rate of aging. So we were interested in whether it was possible to detect variation in the pace of aging as measured by this DNA methylation index even earlier in the life course. And here are data from the Texas Twin Project. These are children who range in age from about 8 to 18 years, but most of them are in this middle childhood range, mean of 13 years. And what we see in these young people, this time we're looking at DNA methylation data collected from saliva, is that kids growing up in families with more socioeconomic disadvantage, as well as in neighborhoods characterized by more socioeconomic disadvantage, again show evidence of this faster pace of aging. And it's worth noting that DNA methylation measures of age, at least, record differences accumulating from the very beginnings of life. So there's a figure from Horvath's original 2013 paper on the left-hand side, and from the Kobor Lab's more recent report on the right-hand side, illustrating that we detect differences, or differences in DNA methylation are detectable really from the very beginnings of life and accumulate with advancing age going forward. Whether these accumulation of methylation changes that reflect the advance of chronological age are also indicating a process of biological aging is something that I think remains to be established, but it certainly suggests that it's possible to measure variation in the process of aging in very young organisms. So I'll leave you with just a few thoughts about what all of this means, and then I'll get out of the way so you can hear from Vadim. So, I think processes of aging that compromise function and drive disability and mortality are ongoing from early life. So one of the implications there is that we can start to think about early life interventions as having potentially geroprotective benefits. And that's something we're very interested in measuring because I think it can have a tremendous impact on how we evaluate cost benefit in public policy. And then on a more practical level, these molecular measures of the pace of aging make it possible to observe public health relevant processes of aging in real time, including in young people. And that means it's possible to do etiological research into the origins of aging-related disease in young people for whom we may not yet have prospective follow-up to the onset of these late life diseases. It makes it possible to surveil populations, to measure trends, and evaluate changes in the aging rate that may be occurring without relying on mortality data, which have been the traditional data source for those kinds of public health surveillance activities. And finally, these measurements may provide us with what are called surrogate endpoints to evaluate impacts of policies, programs, or therapies that may shape processes of aging and therefore affect risk for chronic disease, disability, and mortality. And finally, there's a link at the bottom there that would allow folks to compute our measurement from their DNA methylation data if that's of interest. I'll share the slides with the organizers so they'll be available there, and folks can also contact me if they're interested. But with that, I'll conclude my presentation and just say thank you to the GSA and to Ron Cohancy in particular for giving us this opportunity. And I'll say I'm very excited to hear what Vadim has to say next. Oops.
Thank you, Daniel. And we'll take that as an introduction to Vadim Gladishev's presentation.
Hello, everybody. Can you see my slide?
Yes.
Okay. Well, it was an excellent presentation by Daniel, and actually, I would say that I agree with most of what he said, but I still would like to give slightly different perspective. So, aging is most often measured by changes in mortality. It is said that something ages if it's more likely to fall apart tomorrow than today. And indeed, if we consider human mortality represented here in blue men and orange women, and in green altogether, we can see that in the logarithmic scale, it grows linearly with age. Yeah, so potentially then we could project this pattern to earlier life and predict when it would intersect and then identify the beginning of aging. Of course, the problem here is that the actual pattern is much more complicated. We have here a bump around the age of 20, and then suddenly in earlier life, this pattern goes back. So we have this so-called U-shaped curve in which early life mortality is actually very high, equivalent to maybe a 50-year-old person. So as a result, if you ask people in the field, when do they think aging begins? We typically get 6 different answers. Some people think that aging begins somewhere here by following phenotypes of aging. Some people think it's at the completion of development around age 20, maybe at the puberty around age 13, or minimal mortality age 9, or at birth, and maybe at conception, which is minus 0.75 years. So when is it? So a typical answer would be something like that. It begins after development, at the onset of reproduction, when strength of natural selection begins to decrease. So let's consider these 3 points: minimal mortality, puberty, and completion of development. First, there are 3 different points. They don't correspond to each other. Also, we have to ask a question, especially in the period between age of 9 and age 13, we see that mortality already increases, even if the organism has not yet reached the maturity, so it cannot reproduce. So if evolutionary biologists would be right that strength of natural selection would decrease, then why would we see an increase in mortality here? What is so? It's actually very strange. Why is it the minimum mortality at the age of 9? So then we ask a question, even more extreme question: who is older, 1-year-old or 5-year-old child? Considering this pattern, because mortality is higher for 1-year-old child than a 3-year-old child, we would think that 3-year-old child would be younger, but this makes no sense in terms of mortality. Of course, 3-year-old child would be older. So my point is here is that mortality might not be the best representation of aging process. And to understand when aging begins, we actually have to ask, what is aging? We have to define what is it. And there are different definitions. Some people think that aging is increased mortality, or maybe decreased fitness, loss of function, process of damage accumulation, or decline in system integrity, or a process of becoming old. Of course, all of these features, they characterize the aging process, but there must be one, the most basic, the most fundamental feature, which would influence all other features. What is it? We have to define it to understand the beginning of aging. So I think the essence of aging is the accumulation of deleterious age-related changes, such as molecular damage. So molecular damage is actually a good proxy for it, but it's just incomplete. So why I think so? Consider a very simple case. There is an enzyme that converts a substrate to a product. So this means that there is a gene in the genome, yeah, that codes for this enzyme. And this enzyme is needed to support that particular function. However, when it comes to biology, I mean, this is a chemical process, but chemistry doesn't disappear when we consider biology. Therefore, there is non-zero likelihood that this enzyme will produce something else. Let's call it damage. And of course, I didn't tell you which enzyme is this. It could be any enzyme. Of course, it applies to every enzyme, and not just to every enzyme, but to every biological function that is used by organisms. So in this case, we could extend this notion, and the damage will be presented by byproducts of metabolism, mutations or epimutations, could be transcriptional, translation errors, imbalance in protein complexes, or imbalance in tissues, cell composition of tissues, or could be all kinds of other known and unknown forms of damage. An important point here is that every cellular function produces damage. There's no purpose in this damage. Organism is not designed to produce this damage. This damage happens as a consequence of this part of the function. So as a result, damage is a kind of a byproduct of being alive. So the term damage is a little bit of limiting. So in the lab, we call it the deleteriome, or maybe it can be represented more by cumulative damage. But the damage itself is a limiting term because it's not just molecular damage. It's really any negative consequence of being alive, any negative or deleterious age-related change. Therefore, it's really a combination of deleterious changes rather than just molecular damage. And all other phenotypes, age-related phenotypes, such as increased mortality or decreased fitness and so on, are the consequence of this accumulation of deleterious changes, such as damage accumulation. So now, instead of mortality, let's look at how damage accumulates as a function of age. And we could do it by quantifying accumulation of mutations. And here we've done it in this paper by following accumulation of mutations by looking at childhood cancers as a proxy of aging in this case. And we can see that there is no U-shaped curve. Damage begins accumulating very early in life and kind of continues here in every case. Also, we can look at epigenetic changes, and here, both in human and in mouse, damage begins to accumulate very early in life as well, apparently before birth. So what this means is that aging begins very early in life. The question is when. To understand it better, we have to go to the 19th century ideas of August Weismann. So he proposed that there is a separation of the germline, which is essentially ageless, and soma, which ages. And we can find this quote literally in thousands of papers, which says germline does not age. And of course, it's true in a sense that if germline would age, then each next generation would start at a higher biological age, and eventually the entire population will be extinct. So germline or the organism has to start aging at the same level in each generation. However, germline also accumulates age-related damage over time, such as byproducts, epimutations, modified proteins, which means that it becomes older over time. So for new life to begin in the same young state, the germline must somehow be rejuvenated. Its biological age must be decreased somehow. So the idea then is that the germline ages during development and adult life and is rejuvenated in the offspring after conception, because that's the only place where it can rejuvenate. So this leads to a model in which initially here at the level of zygote, immediately after fertilization, the biological age is not minimal. It's actually relatively high, and then it decreases to a particular point, to a minimal during embryonic development. Let's call this point ground zero. And then it begins to increase again, and it increases for the rest of life. So this idea of this ground zero in the mid-embryogenesis, or maybe a little bit in the earlier part of the embryogenesis, is kind of related to the evolutionary hourglass model, which is illustrated here. You see, across vertebrates, at the level of zygotes, there's a great diversity. So we can easily distinguish like a zygote from the turtle, from the zygote of a chicken or mouse, and so on. Yeah. But then during development, all these organisms, they acquire a very similar state in which you can barely distinguish like chicken from a turtle or from a mouse. And then they diversify again. When the vertebrates are born, they are quite different here. So this state is called the phylotypic state, and it's characterized by the use of evolutionary oldest genes and also by constrained gene expression. So what we can say that if this state is related to the ground zero state, this means that aging begins at this ground zero, and actually organismal life begins also at that point, in contrast to cellular life. It is characterized by the lowest biological age, and as well as by the expression of the evolutionary oldest genes, and by constrained variance in gene expression. So how can we test this model? So we could use, for example, epigenetic clocks or some other clocks that quantify the aging process. For example, if you build a clock by using this red dots, this data, and we would collect samples and build a clock, and then we could project how it behaves in this direction, in the oldest direction, as well as in the youngest direction. If there's a steady increase in aging from the very beginning, we would expect a pattern like that in blue. If the pattern, if there's minimal here during low early embryogenesis, the ground zero here, we would expect a pattern like this. And the prediction would be that there should be a period of rejuvenation between the conception and ground zero, and that cells corresponding to early embryogenesis should not age. Cells which are here, if they would divide, they should not age. Let's test this idea or test these predictions experimentally. So here is a pattern of DNA methylation age during early embryogenesis. 0, which is the conception, and this is embryonic development in mice until they are born around here. You can see that initially the age is a little bit high, or at least it's not a minimal, and then it actually decreases initially and then increases again. So there is a minimum here. And importantly, it does not correspond to the patterns of DNA methylation change itself, because there's a wave of DNA methylation of demethylating the cytosine, and the minimal here actually is much earlier than the predicted ground zero. Also, when we study aging of embryonic stem cells and induced pluripotent stem cells, we also find they don't age. So this is embryonic stem cells using the Horvath multi-tissue clock, and you can see that even if they are passaged over 100 times, over 100 passages, we can essentially see almost no difference here. And this is passaging of induced pluripotent stem cells. Again, many passages. We don't see any evidence of aging in these cells. So therefore, both of these predictions are satisfied experimentally. Of course, this is still a preliminary data, need to be verified further, but at least there is initial evidence in support of this model. What are other supporting evidences? This paper, they found that in mammals, male germline indeed ages based on DNA methylation clock. Again, this would suggest that maybe it needs to be rejuvenated, and when is it going to be rejuvenated? Presumably after conception. This paper, 3 years ago in Nature, finds that germline in C. elegans ages, and they studied protein aggregates. And it was also found that sperm signals clearance of aggregates in oocytes, presumably indicating a preparation phase for this rejuvenation event. Finally, there's this very nice paper, and this paper reported that initial passaging of mouse embryonic stem cells derived from inner cell mass, it supports extension of telomeres. And if we take these cells, which are presumably rejuvenated, and make mice from these cells, the mice live longer than the mice made from ES cells which were not passaged initially. Again, suggesting that cells were rejuvenated during this initial event, and then therefore rejuvenated cells supported longer lifespan later in life. So if what I told you is true, we could better define the relationship between aging, development, and rejuvenation. So what is aging? Aging is basically an accumulation of damage and other deleterious changes that leads to a higher biological age. It begins at ground zero in mid-embryogenesis, ends when organism dies. It has no purpose. On the other hand, development is a genetic program. It has a purpose to build a fit organism. It begins at conception and ends when around 20 years in human. So it's often discussed that aging is a continuation of development. However, this model suggests that aging and development, they proceed in parallel, and aging, at least during this period, is a consequence of development, is unwanted byproduct of this genetic program. Rejuvenation that then would be a removal of damage that leads to a lower biological age. So natural rejuvenation it begins at conception and ends at ground zero, when we see an initial kind of decrease in biological age. But of course, other examples of rejuvenation is the conversion of fibroblasts to induced pluripotent stem cells. So what this means is that Weismann's immortal germline and aging soma ideas are of course correct, but they may be extended by suggesting that actually both soma and germline can age, and they both can be rejuvenated, both naturally and in a test tube. So finally, the question when aging begins, My answer would be that at the embryonic ground zero, it's right here. It's not at the very beginning of life, but it would be slightly later when this cell division would support rejuvenation and removal of damage. And then aging would begin here. It might correspond approximately to phylotypic period, although we don't know for sure. And then it would increase and increase, and this would be the birth, and then it would continue increase and increase until organism dies. And mortality is a late manifestation of this process. We only see it at the age of 9. So age 9 means actually it has no particular meaning. It just happens that there are two processes of early life mortality, and the aging process overlap at the age 9. So what I told you is described in these 3 papers, and in addition to these authors, preliminary data we obtained by Csaba Kiripesi, Bohan Zeng, and Senggu Li in my lab, and all of these studies are supported by NIA. Well, thank you.
Thank you, Vadim. Outstanding presentations, both. I'm going to begin with 1 or 2 questions, and I'll start with a question for Dr. Belsky. In epidemiological studies, I think one of the questions in starting with an older population would be inclusion and exclusion criteria. And is it not possible that in doing a study starting from older populations, there's some bias in how the population is selected that might prejudice the kinds of conclusions about rate of aging or pace of aging?
Oh, it's a great question. It's something that we think about a lot. It's one of the reasons I think it's so important to begin building models of aging with relatively young populations if we're studying humans. The argument is that as we get older, our risk for death increases. So if we think about a birth cohort, you know, nearly all of that birth cohort is still alive 20, 30, 40 years later. If we're talking about humans, there's some selective mortality, but most of that selective mortality is not due to causes that we would relate back to biological processes of aging. In contrast, if we go a couple of decades forward in the lifespan, now we're talking about seeing only, let's say, 70, 60, 50% of that original birth cohort, with the loss to follow-up being largely due to what we presume are outcomes of biological processes of aging. In other words, the chronic diseases of aging that are causing death in people in their 50s, 60s, 70s, and 80s are arising, at least in part, from the biological changes that we're trying to study. And so when we use as a proxy for an aged human, the average 80-year-old that we observe in a cohort study, we're actually looking at what must be, by construction, a relatively slow aging member of that original birth cohort. And if we're using those individuals to parameterize our models, whether they're physiological models, molecular models of aging, it may bias those estimates. And this is something that was actually addressed in a recent very thoughtful commentary in the Journals of Gerontology. I think that it's also the case that when we include a wide age range of individuals in our studies, we're combining not only information about aging, but also about exposure histories that vary by people's birth cohorts. And so an individual born early in the 20th century grew up under a very different set of pathogen exposures, had a very different set of nutritional exposures, very different set of environmental toxicant exposures than someone growing up at the end of the 20th century. And those differences, while they may ultimately impact aging, are not themselves the substance of biological aging. And so we need to think about how we can control for those cohort differences as well. Okay, so that would be my answer.
Thank you. Okay, I will ask the following question of Vadim. So during embryogenesis, especially in mammals, there's a high failure rate. Would you, and embryos die before they come to term, So that death is different in your scheme. I think that death is different from death after birth and in older ages. Once you get past the puberty, past the adult development, and they're reproductively competent adults. So embryonic death would not be an index of aging in the well. Death is not an index of aging anyway, but the death is a different result. It's not a damage accumulation death, or is it? So what I'm asking is, there's damage accumulation. Does it have the same consequence vis-a-vis how we think about aging during embryogenesis versus adult life?
Yeah, Ron, thank you very much. This is an excellent question. So indeed, we have to ask, what is the cause of this early life mortality? So clearly, it's not aging. So initial mortality is high and then decreases during embryonic development and all the way to the age 9. So what are the causes of this mortality? So one cause is embryonic failure rate, failure, and so developmental errors. That's very clear. But there is a second very important cause, and this cause is the kind of early life selection. So what I mean by this is that there is a offspring inherits genotype from parents, and if parents have a high rate of many damaging mutations, the offspring may get disproportionately many such mutations, and then the offspring would not be viable and would die during embryonic development. This allows for damaging mutations to be removed from the population. So when I discussed early life rejuvenation from conception to ground zero, of course, during this period, cells, they divide. They need to clear damage by diluting it and repairing it. For example, they increase the telomere length during this period. They demethylate the epigenome. They have some other manipulation. So a lot of things can be rejuvenated. On the other hand, the genome cannot be, because we have genome fixed. We have 50% of the genome from mothers and from mother and 50 from father. And in each generation, we accumulate more mutations. So each new person has on average 70 new mutations. So somehow these mutations, they need to be removed from the population. And this early life mortality is one opportunity. In essence, we rejuvenate the genome. You see, we rejuvenate molecular features by cell division, and we rejuvenate the genome by this early life mortality. So I think this is not very well understood at the moment, and there are some studies, but I think further studies need to be done just to verify this. But we see later in life, or later in childhood, this at the level of autism. For example, autistic children, they inherit more mutations from parents than their healthy siblings, but it's just probably represented by to the more extreme by early life mortality during embryonic period.
Okay, thank you. I'll go back to Dr. Belsky. So do you have any evidence that earlier in life there's more damage accumulation in low SES and less damage accumulation in high SES humans?
So I think to that question, we're observing some evidence of this faster pace of aging in these kids, which could be sort of one piece of evidence toward that question. Is there more damage accumulation? We also know that kids growing up in lower socioeconomic status households are exposed to more damaging agents. So they have higher burdens of environmental toxicant exposure. They have higher burdens of chronic psychological stress. They may have poorer quality nutritional environments, and so in other words, both physical and psychological dimensions of their environment contain more damage-causing elements, and we observe in these indirect measures of aging signs that their bodies are aging more rapidly, and I think those together sort of triangulate toward this answer to the question that you're asking. Ultimately, I suppose we would need, to generate evidence along the lines of Vadim studies, high-depth DNA sequencing in kids from these different groups of families to evaluate, for example, burden of somatic mutations accumulating in different kids.
Okay, thank you. Back to Vadim, I have a quick question about using cell passage number. So earlier in embryonic development, is not the rate at which cells cycle somewhat faster than later in embryonic development? And so would that influence the appearance of more cell passages from earlier, before the ground zero state is reached?
It may be. We are not sure about this. Apparently, cells may exist in the state when dividing, they accumulate damage. For example, fibroblasts, when they grow in culture. There are some situations when cells, when divide, they decrease damage, like, for example, during early embryogenesis. And there are some states in which cells, when divide, they maintain a relatively stable level of damage. For example, iPS cells, or perhaps cancer cells, some of the cancer cells. So what defines these differences in the cellular state? It's still unclear, but I think it would be an important point of investigation.
Okay, on that, Hanum and study came out about the same time as Horvath's from Eidecker and Zhang labs. When they looked at tumor cells, they found that they appeared to be younger in terms of their methylation state. So that would be consistent.
Yeah, it would be consistent because there are different cancers. Some cancers presumably might actually become younger a little bit, and some than the organism, and some would be older than the organism, and some would be the same.
Sure.
Yeah.
Okay, so I'm going to turn to a couple of questions from people who are listening. This one is, I don't know if I'm allowed to read the name, so I'll just read the question. I'm curious to hear more about aging as having no purpose, quote unquote. Could the argument be made that if the purpose is to perpetuate the life cycle, in other words, life needs both a beginning and an end? It's almost T.S. Eliot. So just asking you to expand on the comment, aging has no purpose.
Okay, I guess it was my statement. Well, this referred to the program theory of aging, although I didn't mention it. Same August Weismann actually in the 19th century proposed that aging is programmed. There's a purpose in aging, that that the organisms would die to free space for the new generations. However, based on what we understand now about the aging process, aging has no purpose. It's not programmed. It appears programmatic to some degree because damage is mostly produced by genes, and genes are encoded in the genome. So it's not just random damage, not really stochastic damage to a large degree, but a particular damage produced by particular enzymes and particular biological systems. So the damage produced by enzymes, for example, in yeast, would be somewhat different from the damage produced in the mouse, because different genes produce it. So it's not really random. So, but at the same time, aging, yes, it's programmatic, but it's not programmed. There's no purpose in it. There's no need for the organism to die. It's unfortunate byproduct, unfortunate consequence of being alive. We're metabolically active and produce damage. And damage accumulates, organism dies. So that's my answer.
Okay. I assume that people can see the questions and answers as they're being posted online. But in the event that people are not looking, there is a question which often comes up: why is aging characterized as decline and not accretion as in wisdom? Daniel.
I think this is a great question, and it's one that I often try to address at the beginning of my talk, and I think this time maybe I skipped over it. But to me, this is very much a question of perspective. As humans age, we experience all kinds of changes. Some of those changes can be characterized as growth and development, and there's a whole field of adult development concerned with the study of those changes. Research into aging biology, in part because it's based in yeast and worms and flies and mice, doesn't think very much about the subjective experience of aging from the perspective of those agents. And as we translate it into research on humans, we're very much concerned with understanding the causes of and preventing diseases and disabilities that shorten healthy lifespan. And so the biological process of aging that we're studying is very much one of decline. It's occurring at the same time, though, as the questioner notes, as this other set of changes that may be understood as growth and development. And so I think it is an interesting question to ask: how do these individual level processes of growth and development that occur with advancing age interact with this ongoing accumulation of damage and decline in system integrity caused by it?
Thank you. So I have one question for both of you, and I hope I can phrase it in a reasonable way. So damage accumulation is one part of the story, and a major part, as most investigators believe. But there's also the ability to buffer against damage. So there are a lot of genetic manipulations that in laboratory animals can extend lifespan and appear to produce healthier animals, as well as pharmacological, dietary, or exercise interventions. And also in human populations, we know the effects of good diet, moderate exercise, and some suggestions of anti-aging therapeutics, which may be tested. So would you comment on whether buffering against damage is a likely factor in some of these laboratory studies, and how would you assess that? Also in human populations, what would you expect to see? Start with
maybe
or Daniel. Sorry.
I was going to say, I think Vadim is going to have the better answer to this question, so maybe let's let him have the last word. What I would say is that, to the extent that we're thinking about this question, it's very much in terms of applying quantifications of biological aging as surrogate endpoints in trials of therapies or evaluations of programs that we think may induce that kind of buffering. So for example, the calorie trial, which prescribed 2 years of caloric restriction to healthy non-obese humans, is one of the first human geroprotector trials, and there we're testing to see if that intervention, which we know slows aging in worms, flies, and mice, is also doing so in humans. But we're again asking this question of mechanism only indirectly via these existing measurements of biological aging.
Vadim?
Yeah. So my thinking is that, yeah, this is an important aspect of aging. When damage accumulates, organisms have to respond to it. So they do respond. They respond metabolically or in the form of gene expression or genetic changes. So basically, age-related changes, they involve accumulation of damage as well as response to this accumulation and damage. Therefore, it's actually very challenging to infer causal relationship from the age-related changes, because we don't know whether these changes are negative, positive, or neutral. So they can be used to quantify the aging process, for example, in the form of DNA methylation clocks, but in terms of how these age-related changes influence the organism, in which way, it's very hard to say. But basically, yes, it's a damage and adaptation to damage. Adaptation could involve protective systems, of course, and as well as other changes.
Okay. Excellent. Very provoking, which is the intention of thought and possible experimental approaches, some of which have been outlined in both human studies and animals. So the next in this series will be Alyssa Eppel and Ann Newman. That will take place November 23rd from 3 to 4 p.m. in the same format as we have here. Thank you, gentlemen, for very stimulating presentations and taking the time to engage in this. And thank you to Judy Liu and the Gerontological Society of America for making this possible. You have my deep appreciation for helping out on this. Thank you very much.
Thank you, Ron, and thank you, Judy.
Thank you, Ron. Yeah.
Take care.
Thank you so very much. Bye-bye. Big thank you to the panel and to our participants. This is the end of the program.